All right, everybody, I think we could go ahead and get started. Thanks so much for coming out tonight. Super excited to welcome our guest speaker, Sarel Fleishman. Sarel's in town, some pizza and drinks. Sarel's in town this week for PEGS, and so we were super lucky to be able to line his schedule up to come speak at Protein Design and Modeling Club. But before I introduce Sarel, just want to say big thanks to all of our sponsors. They're the ones that pay for the pizza and drinks. And so very grateful for all their support. For the folks in the audience that work for like a big pharma, please go and talk to the folks there and see if they'd be interested in getting, supporting the group here and getting their logo up. This is how we keep it going. So, we also rely on our members to help us support this. So yeah, thanks everybody. All right, so Sarel is a total boss, and we are super lucky to have him here. So my first introduction to Sarel was actually while I was interviewing in the Baker Lab, and I think that was when the first ever design of a protein-protein interaction. And I was interviewing with David, and Sarel comes in and has the manuscript revisions for the Nature paper in his hand, and it just melted my brain. It was amazing. And so this is Sarel's career has just been a rocket ship. Not only is the stuff really innovative, but he also makes it really accessible. Things like PROS, where you can take proteins and have a reliable web server to make them more stable. And there's just been innovation after innovation like this. So Sarel is also now becoming very entrepreneurial. So founded and is the chief scientist of Scala Biodesign, in addition to his role in academia. And so, yeah, really grateful to have him here tonight. And Sarel, please take it away.
Thank you very much, Chris, for this lovely introduction. I didn't even know about this, the first meeting. Let me get out of this for a second and into my own presentation. Just a second. Yeah, all right. Okay, now we're ready. And this, all right. Yeah, okay. So again, thank you very much. So the goal of the research in my lab is to understand how protein function is encoded in sequence and structure, and to translate this understanding, as Chris said, into practically useful tools that anybody can use in order to optimize stability and activity in natural proteins, or to design completely new function. So to say the obvious, programming activities in proteins is really what we all care about. We'd like to be able to decide on a function and to be able to design that function in proteins. Let me see that I can make this work. Yes, but there have been persistent challenges and barriers on the road to designing functional proteins. This is taken from a very recent review. Sorry about that. Okay, yes. Taken from a very recent review from David Baker, Dan Hilvert, and Anthony Green in Nature, where they reviewed the progress in de novo enzyme design. And after reviewing this progress, they say we must learn how to design highly active enzymes with efficiencies more akin to natural systems. And the reason they say this is that all de novo designed enzymes to date, even today, this is from 30 years ago, have been orders of magnitude lower, or have shown orders of magnitude lower catalytic rates than comparable natural enzymes. Now, why is that? What are the reasons that people, including in this review, have brought up to explain these huge gaps between natural and de novo designed enzymes? These have been the low stability and expressibility of designed proteins. There has been also, we've seen that low activity may be due to inaccurate design of active site constellations, and also inaccuracies in backbone design. So in order to solve this challenge of how to program completely new activities in proteins, we need to figure out a way to solve not just one problem, but all of these three problems. So essentially, the bottom line of this is that despite huge progress in de novo design over the past 2 decades, fundamental problems, including foldability, stability, and activity, are still incredibly challenging to design. So the defining question for our field, this is my sense of it at least, is how do we design highly functional proteins completely computationally without using in vitro evolution. Okay, this is what we're trying to achieve: completely computational, completely programmatic design. So, needless to say, I think in this room there has been huge progress in fold design over the past decade, and even this slide from just a year ago is already outdated by contributions from Sergey and others, which have gone well beyond what we see in this slide. But even with these contributions, what we still see is that de novo designed folds are quite simple in terms of their topologies. They mostly comprise of tightly packed alpha helix bundles. By contrast, the proteins we care about most in terms of functions, like antibodies, enzymes, and vaccine immunogens, are remarkably complex in terms of their topology. And this complexity of topology is not just a bug in how enzymes and antibodies are constructed. These are features. These are responsible for the activities of enzymes, antibodies, and other proteins. For instance, the cavities we observe in enzymes, these are the active site cavities that are exquisitely structured to recognize their substrates. Without these, enzymes would not be as efficient. So natural proteins provide a treasure trove of molecular functions. But these structural complexities, these non-ideal structural features come at the cost, of course, of low stability. And of course, natural proteins they don't exhibit the functions that we care about. So the way that people have been optimizing natural proteins, also de novo designs, is by using the Darwinian paradigm of trial and error. We start with some low level of the property we're interested in. This can be the stability of the protein or the activity levels of the protein. And we start introducing diversity. This can be by random mutagenesis or more systematic approaches. Be that as it may, by introducing just one or even a handful of mutations, progress is not very high, right? We need many mutations in order to achieve large progress. And this means that we need to iterate this process of mutation and screening, and this leads to lengthy cycles that last months to years to PhDs. But of course, even when this process converges, we may find ourselves in these low-lying hills and on Fleishman's landscape, because one property that we're interested in, let's say the activity of the enzyme, even though we've been able to optimize it very far, another property that is absolutely essential, such as stability or expressibility, has deteriorated. And these activity stability trade-offs and other trade-offs are incredibly frustrating. They can completely block the optimization workflow. But beyond these practical concerns, what I feel most passionately about is that the fact that we still rely on chance in order to generate life-saving drugs and enzymes for industry and for research means that there are remarkable gaps, large gaps in our understanding of how function is encoded in proteins. And the goal of our research is to try to minimize these gaps as much as possible and to enable what we call one-shot, non-iterative protein design, introducing as many mutations as needed in order to reach the functional goal without observing trade-offs. Okay, that's our goal. So I'll start by describing several of the methods that we've designed or developed over the years in order to address the problems I showed earlier, which are, again, expressibility and stability, activity, and foldability. And then I'll go to new and unpublished work on de novo design of enzymes. So the most general problem in protein optimization is the fact that natural proteins tend to be only marginally stable. And because of this marginal stability, of course, natural proteins exhibit limited usefulness. So it turns out that more than 50% of non-membrane proteins in any proteome tend to be difficult or even impossible for heterologous overexpression. So what we're looking for is a way to stabilize proteins, but without reducing their activity. Again, these trade-offs, we don't want to see any of these trade-offs. And what we're looking for are general rules that can be universally applied to any protein. We're not looking for a specific strategy to optimize, let's say, even an important class of proteins like antibodies. We're looking for general rules that can be universally applied. If we're looking for general rules, we need to think about this through the most general and fundamental aspect, which is, in the case of proteins, the folding landscape. So, of course, you may remember these from basic biochemistry or biophysics classes. According to the thermodynamic hypothesis, the native state of the protein lies in a low energy, low free energy minimum compared to any of the misfolded and unfolded states. This is according to the thermodynamic hypothesis. But for large and complex protein, and literally any protein we care about, so the average protein of the human body is 350 amino acids. So every protein we care about is large and complex. There may be many such misfolded states that can kinetically trap protein tries to fold into the native state. That can lead to misfolding, loss of function, aggregation, bad properties. So our goal in design of stability is to minimize the energy of the native state and to eliminate as many of the misfolded states as possible and accentuate the energy gap between the native state and any of the remaining misfolded states. But herein lies the conundrum for any atomistic design strategy, because the only structure we actually know anything about in terms of atomic detail is the native state. We have absolutely no information about any of these misfolded states and no access to such information. We simply don't know what they look like. So how can we develop an atomistic strategy to counter states for which we have no information? This has been, I think this is the major conundrum for any atomistic design strategy. And we were wondering about this for many years. How do we address this challenge, which is essentially a negative design challenge? But fortunately, we know that mutations that cause misfolding are purged by selection. And that means that there's an easy, potentially an easy strategy to counter these misfolded states. We can simply look at the evolutionary record and sequences of homologs of proteins that we're interested in. Let's say we have a query protein, we look for homologs of this protein, and at each position, we can build a statistical model of substitution in evolution. So essentially a conservation pattern at each position. And mutations that are rarely observed are likely to lead to either, of course, they can also lead to dysfunction, but they're likely to have led to misfolding, aggregation, and all the properties that may lead us to these misfolded states. So in addition, our solution to this problem of how do we minimize native states and eliminate misfolded states is to stabilize the native state while disallowing mutations that are rare in the evolutionary record. Okay? So we're combining here Rosetta atomistic calculations and phylogenetic analysis based on a sequence alignment of homologs. Does this strategy work? Apparently it does. This is how we developed PROS, protein repair one-stop shop. And after we had shown that this strategy works for a number of very challenging enzymes and binding proteins, we wanted to see whether it could also address a contemporary biologics optimization problem. And so we turned to the problem of malaria vaccine immunogen design. So malaria is still a very serious disease. It causes more than 600,000 fatalities per year, mostly of young children in developing countries, for instance, in Central Africa, but also in Southeast Asia and South America. And despite 60 years of research, there's only one vaccine that exhibits limited efficacy, and it won't lead to the eradication of this terrible disease. But this protein called RH5 is considered to be the primary candidate to serve as a complementary vaccine to the one that's currently being used. This is a vaccine for the blood stage of the malaria parasite. And we know that this protein is absolutely essential for the process by which the malaria parasite, the Plasmodium falciparum parasite, invades our red blood cells. And we also know that antibodies that target this protein can block malaria invasion. So this protein is the primary candidate to serve as a vaccine for the malaria blood stage, but it's very challenging to develop as a vaccine candidate. It exhibits limited stability. It starts losing activity at about 42 degrees Celsius. And this is a problem for a vaccine or a protein intended for use in Central Africa, where there's no electricity and therefore no cold chain of transport. The second problem is that the topology here is so complex and the protein is so unstable that it requires very expensive insect cell expression systems. And those are very difficult to use in mass production. So, and of course, that would raise the cost of producing this vaccine. And keep in mind that the vaccine for the developing world, this means that we would need probably billions of doses to deploy for this to be effective. So vaccine requirements, to make this into a feasible vaccine, we need a cost-effective, likely microbial expression system and stability to go well beyond 50 degrees Celsius. Another challenge here is that this is a novel fold. This only exists in malaria, And when we looked at this and searched for homologs, there were very few, fewer than 10, very distant homologs. So this would be impossible for any data-driven method. So here we use PROS to design 3 variants of this protein. Each of them, PROS tends to introduce dozens of mutations to the protein, simultaneous mutations. We just test these mutations. And we typically, with PROS, test between 3 and 5 different constructs experimentally. So this is what we did here. We sent 3 designs to our collaborators in Oxford, Matt Higgins and Simon Draper. They're the labs that are advancing RH5 towards clinical use. So RH5, the wild-type RH5, simply does not express in bacterial cells. There's no expression at all. This is in Western blot, so if this indicates that there's absolutely nothing there. By contrast, the 3 designs we suggested, again, dozens of mutations from the wild type, they exhibit robust expression in microbial, actually standard bacterial cells, E. coli cells. And that already reduces the cost of production by orders of magnitude. This is a very good sign. They took design 1 forward and showed that its activity is retained up to 55 degrees Celsius, which means that potentially this design would not require a cold chain of transport. Again,
can you comment a little bit about why you think they expressed in the bacteria?
Yes, yes, of course. So this is something that we've seen time and again with PROS. It's not guaranteed to work this way, but bacterial cells lack the chaperones that we have. So bacterial cells have a handful of chaperones, whereas our cells have many, many hundreds. And that means that or eukaryotic proteins tend to be less stable than what we see typically in bacterial cells. So if we want to heterologously overexpress a eukaryotic protein, and this is true for pathogens, the problem is even more serious. They tend to be even less stable. In microbial cells, we tend to see problems. So it's a question of proteostasis, essentially. So, and now why do the mutations improve stability and expressibility? The fact that you reduce the energy of the native state and you essentially smooth the folding landscape means that the protein tends to the native state more easily, and it requires less the chaperone network, the proteostasis network to fold. That's the hypothesis, at least. It's very difficult to prove, but that's the hypothesis. All right? Yes?
I guess you're going to talk more about how PROS works, but earlier you were introduced. I guess you're going to cover on what exactly PROS as a method does, but earlier in your introduction, you were explaining how you can capitalize on evolution and mutation. You kind of contradicted yourself with saying that this is so distant and very few homologs exist.
Yes.
So how do you explain that?
It's true. So let me I won't actually provide more details on PROS. The papers are published, but just explaining what we do. Essentially, we generate the multiple sequence alignment of homologs. In this case, there were very few homologs. So I'll explain what I think is going on in this case. And from this multiple sequence alignment, we generate a standard PSSM, a position-specific scoring matrix. That's essentially a statistical model of substitution in evolution at each position. So it gives you essentially the log likelihood of mutation, each mutation at each position. And we translate these log likelihoods into Rosetta energies. And we combine this into the Rosetta energy function. This is essentially what happens in PROS. It's a very simple idea of taking the probabilities from natural evolution, transforming them into Rosetta energies, combining that into one energy function, and then optimizing of course, there's many details behind there, but all of that is in the papers. Now, it is true. So when we published PROS initially, in the limitations section, we wrote that we would likely require dozens, if not hundreds, of homologs to make PROS work. And the second example was this RH5 protein, where we had 7 distant homologs. Yeah, I think that what we need is even some information from nature about what is allowed and disallowed. And that in itself is already sufficient, which is, I think, remarkable. Yeah, it's just a second. It means that the Rosetta energy function is fairly close to being realistic. And what you need is a little bit of guidance. I think that's the correct interpretation of this. Yes, please?
Well, we are on the topic about language.
Excellent, yeah. Yeah, so what about protein language models? I was hoping that they would work. I don't think so anymore. We've done so many tests on protein language models, but we do have a recent paper on a completely different, again a malaria protein, but from vivax, where we wanted to stabilize it. And in that case, homology again, where there were so few homologs, process simply didn't work well, and we used MPNN to generate the PSSM. So MPNN is not a language model. But the fact that that protein is sort of like an alpha helix bundle suggested to us that MPNN would work well on it. And indeed it did. So MPNN was used to produce a PSSM, then that was thrown into Rosetta as a standard PSSM in PROS, and that worked very well. But language models, we're not giving up, but we're really discouraged by what we see. We don't see evidence that they work for protein design.
You mean when you don't have homologs? Because when you have homologs, they essentially produce the same.
Exactly, that's true. But when you don't, yeah, so I'll just repeat that. When you have many homologs, they reproduce the distribution of mutations that you see in nature anyway, so a PSSM would be good enough. When you don't have homologs, sorry. Yeah, I was hoping, I was really hopeful about that. Yeah, sorry, yeah.
Did you do anything specific to keep the binding sites for the neutralizing antibodies?
Yeah, very good point. So in PROS, even the web server, which is shown there, so it asks the user to input what are the sites, the active sites, if this is an enzyme active site or a binding site. And here, as you said, we input not just the neutralizing antibody binding sites, but also the receptor binding site on RH5. Thankfully, we had structures for all of that. Yeah, that's a really good point. So the protein is much more stable. Then our collaborators, again, took this into animals and showed that despite these dozens of mutations in this design protein, the neutralizing activity, the neutralizing activity of the antibodies they extract from animals is essentially the same as for wild-type RH5. So that was very promising, but I'd like to focus on more recent results. This is from last year, this paper from last year, where essentially what they wanted to do is to boost the immunological efficacy of the protein. So they wanted to load RH5 on virus-like particles to increase the avidity. Avidity tends to go together hand in hand with increased immunological activity. But RH5 is so unstable that it couldn't be loaded efficiently on virus-like particles. By contrast, our design could be. And that led to an order of magnitude increase in immunological response. And that's really, I think, a beautiful result, showing how stability can translate into a better construct for immunological activity. And based on these results, this protein entered phase 2 clinical trials in West Africa, including young children last year. And I find this to be, of course, very, very exciting that we may be able to contribute to eradicating this terrible disease. And just note that this is, as far as I know, the first example of a completely automated biologics optimization algorithm that has come so far in usability. And I'm sure that there are others that will follow this. So everything that I'll show you today relies on having an accurate molecular structure. And until recently, that meant crystallographic structure. We couldn't even use NMR, and cryo-EM was also suspicious in these cases. But of course, with the advent of AlphaFold, we asked ourselves whether AlphaFold models, of course, they're not as good. They're not as good as crystallographic structures, especially in the active site. We wondered whether they would provide sufficiently accurate models for PROS and other methods that we developed. And the answer is largely yes. This is, I think, a very striking example. Here we took a family of enzymes. They're called unspecific peroxygenases. These are enzymes from fungal sources, and they're very difficult for heterologous overexpression, even in yeast. Yeast are homologous to these to the source fungi, but even yeast cannot express most of these UPO enzymes, and they're very interesting for sustainability research and for medicinal chemistry purposes. So our collaborator, Martin Weissenborn of Halle University, suggested that we work on 10 different UPOs that they were never able to express in the lab and characterize them. We generated AlphaFold models for them. We had to adjust PROS, and this is now automated on the web server so that it can deal with the inaccuracies using PLDT scores. The inaccuracies in AlphaFold models But basically, that's all we did, a standard PROS strategy. Each such design encoded about 40 mutations relative to the wild type. But for 9 out of these 10 targets, we got highly expressed and secreted and functional UPO enzymes with high stabilities. And finally, they could characterize their activities. And of course, they observed different activity profiles for these enzymes, showing potentially a path for the discovery of new functions in natural enzymes. And in addition to that, recently we extended PROS to membrane proteins. Of course, the membrane proteins reside in a completely different environment from soluble proteins, so we had to adjust the Rosetta energy function for that, but it worked very nicely. This is for a voltage-gated potassium channel, which is a very dynamic protein, and yet PROS worked. Activity was maintained completely, but expressibility in oocyte cells increased by 14-fold, if I remember correctly. And this is for a human enzyme, membrane enzyme, again, of unknown structure. So again, based on an AlphaFold model, again, expressibility increased, activity was maintained. This is available also by web server. So of course, although we had to change the protocol in some aspects in these different design scenarios, the same principles that I showed in the first slide of this section are relevant in all of these cases. And this suggests that the stability of design principles that we encode in PROS may extend to all proteins. And given the fact that really developing a general or universal strategy for stabilizing proteins has been a very long-standing, decades-long challenge in protein science, I think this is very encouraging. So the next question is, as somebody pointed out here, in PROS, we're only designing the scaffold of the protein away from the active site because we want to maintain activity. But what if we want, the question is whether we can extend the strategy in order to improve the activity. And that means that we're going to target the active site pocket. If this is an enzyme, it will be the active site pocket of this enzyme. The problem with this is that active site design impacts the most sensitive region of the protein. Right? This is the region of the protein that's actually responsible for function, and mutations in the active site tend to cause loss of function. And this is the reason why active sites tend to be the most conserved parts of the protein. Let's look at the details of an active site to try to understand why it's such a constrained problem. So this is taken from glycoside hydrolase enzyme. The ligand here is a xylan polysaccharide. And as you can see, and this is true for almost any enzyme, the enzyme active site pocket residues form a very dense interaction network with the ligand, many polar interactions with the ligand. In addition to that, and what sometimes people miss, is that the active site residues also form a very dense network of interactions with one another. And you can imagine that introducing even a single point mutation in this dense network, let's say to the tryptophan, would cause the collapse of all the other residues in the pocket, leading to orders of magnitude decrease in activity. And this is essentially the reason why active sites are so conserved. How does evolution deal with such a problem? It would have a very hard time, right? Because each point mutation is likely to lead to loss of function. How would it increase? But we thought, and of course this makes sense, that instead of introducing one mutation or trajectories of single point mutations, what if we could reintroduce constellations, new constellations of residues, let's say 3, 5, 6 simultaneous mutations in the active site pocket that would maintain the stability of the constellation, but change selectivity and substrate recognition. Could that be done? Of course, the reliability of the design method has to be very high to achieve this strategy. So we developed a method called FunkLib, and the first target system for this was a bacterial enzyme called phosphotraesterase, or PTE. This is a very broad spectrum hydrolase. Its native substrate is paraoxin. This is a pesticide. And the hydrolyzable bond is here. But in addition to this pesticide, it also hydrolyzes a range of esters and lactones. The reason it's interesting, though, is not for these substrates, but for these substrates. These are organophosphorus nerve agents. Anybody who's been following international news, VX, Russian VX, Sarin, Soman, these are compounds that are used by certain governments or terrorist organizations to kill off people they're not happy with. For instance, Navalny, so he was probably attacked by one of these compounds. So terrible compounds, they're actually the most toxic to human chemicals we know. One drop on the skin of an adult kills within minutes. So an effective countermeasure for these substrates is urgently needed. And PT does hydrolyze these substrates, but at catalytic rates that are orders of magnitude too low to serve as an effective therapeutic. So there's been a lot of interest in optimizing PTE for these terrible substrates. Let me go back to the slide again. What we wanted to do is optimize PTE for these substrates, but as you can see in this figure, they're very diverse, and also they comprise many rotatable bonds. In addition to that, we don't have a good model for the transition state. How does the enzyme actually work on these substrates? We didn't have a good model for that, and that's actually quite typical. We often don't have good models for the transition states for complex reactions. So here we ask the question whether we could optimize function without targeting a specific substrate. And that often leads to raised eyebrows, like how would you do that? Like how would you use computational design? It seems like an ill-posed problem. How do you use computational design to optimize for an unknown substrate? So here we went back to the basics of what it means to be an efficient, high-efficiency enzyme. And we know that not all enzymes, of course, are high-efficiency, but high-efficiency enzymes tend to be stable, and that's okay. We can use PROS for that. And in high-efficiency enzymes, again, not all enzymes exhibit this property, but the core catalytic residues are pre-organized for activity. And this is a key point. So the idea behind FunkLib is that we're not going to target a specific substrate. We're going to do the modeling entirely in the unliganded state, but we're going to keep the core catalytic residues completely as we observe them in the target enzyme and all of its homologs. So how does FunkLib work? We're keeping the core catalytic residues, which are shown here in gray, exactly as we observe them. We don't allow them, even if this is a histidine, for instance, we're not going to allow it to even rotate. Okay, of course, we're not allowing a change in identity, but we're not even going to allow it to rotate. By contrast, any other residue in the active site pocket, we're going to allow to change while keeping, again, the core catalytic residues in place. And we're going to design between 1,000 and 1,000,000 diverse constellations of active site residues without modeling the substrate. Again, in the unliganded state, and we're going to screen them for stability and pre-organization, rank them by energy, and then just choose the best 50 or so for experimental testing. Very, very simple algorithm. The working hypothesis here is that active site stability and pre-organization are key to high-efficiency catalysis. That was an hypothesis. We did this in collaboration with Danny Tufik's lab. He was a preeminent enzymologist and protein engineer. When I suggested this algorithm, he looked at me as though I was drunk, but he always tended to entertain us in this regard. So we tested this. We first designed 50 variants of PT, and to our delight, dozens of these variants exhibited higher rates for a variety of different substrates. Here I'm showing only the top two for these 4 terrible nerve agents. And what you can see, this is in log scale. So we're getting to almost 100-fold improvements with D27, but up to 4,000-fold improvements for D28 and some substrates. We then decided to do a second round of design for D27 to try to push this up based on what we saw in the first round of design. We designed about 15 or so additional enzymes, and you can see that we pushed this one up as well. At the end of this process, we got to 4 enzymes that exhibited therapeutically relevant activities for all of the 4 target substrates. This was remarkable. In Danny's lab, they had been working on optimizing PTE for these targets for 8 years, and in that process achieved therapeutic levels only for VX. By contrast, here within 2 months of starting the project, we reached therapeutic levels for all of them. So computational design is clearly more effective than evolutionary strategies, but we began wondering whether evolutionary strategies were able to reach some of the designs that we obtained through computational design. And the reason for that is that when we looked at some of the designs, this isn't true across the board, but when you look at some of the designs, this is a crystal structure of one of the designs in purple and the wild type in gray. You can see that in some of the designs, again, some of the mutations are from small to large, like this histidine to tryptophan, histidine to arginine, and also changing the electrostatic properties of the active site pocket, and they're close together, which would suggest that they would show some interdependencies if this were an evolutionary process. So we tested whether an evolutionary process could reach this 4-point mutant, quadruple mutant. So we turned to mutational scanning for this. We start with a wild type enzyme. This is the activity for one of the substrates we tested. Very low, oh, sorry. So the area of the circle denotes specific activity of this enzyme for the target substrate in this case. Very low activity for this wild type. Then we introduce each of the 4-point mutations of the previous slide. You can see increases in activity. And this one is a really substantial increase, this histidine to tryptophan mutation. If this were an evolutionary path, this would be an amazing first result, right? So the next step would be to recombine these point mutations in order to introduce the doubles. And that's where the frustration begins. All of them are lower in activity than histidine to tryptophan mutation, and 3 of them are even worse than the wild type. This is a fitness value. And yet, if we combine this terrible double mutant and that terrible double mutant, you get the quadruple design, okay? This is known in the field of molecular evolution as an example of reciprocal sign epistasis, where the mutations that are bad singly, when they are combined, they're actually very good. And that rarely, in evolution, you never say never, but these are rarely observed in evolution. And that was, for me, a watershed moment, because I'd been looking for such results for many years, because this shows really in detail that computational design can discover functional sequences that may be inaccessible to evolution. And that's really, I think, a key result, showing that computational design can really improve dramatically and beyond what we can get from evolutionary strategies. So we developed PROS and FunkLib and other algorithms from our lab as web servers. And as Chris mentioned, they've been used by thousands, or they're being used by thousands of academics. And that's been really powerful, because if you follow this QR code, you can find a list of more than 200 publications and patent filings based on these methods. And these are in very diverse areas. And in most of these cases, there were no prior experimental data on the systems that people were looking at. So these were enzymes or binding proteins with no experimental data on mutations. And yet, in all of these cases, they saw dramatic improvements in, again, stability, expressibility, or activity, providing or verifying that these methods enable one-shot optimization of function. Again, these have been in many types of enzymes and therapeutic proteins. And again, as Chris mentioned, this has also raised interest in industry, and graduate students from the lab started a startup company based in Israel called Scala Biodesign that has been collaborating with pharma and chemical production companies in order to translate these methods into practically useful tools for industry. These examples, I often read them in detail because I think we can learn a lot from these examples. I'll take you through one of these because it shows a very nice case where stability design doesn't only translate into higher stability, but also to higher activity. This is for SARS-CoV-2 vaccine immunogen design. And this was done, we weren't involved in this at all. This was done by a group at GSK. So in their paper, in the introduction, they state the following. The identification of an antigen with preferred qualities often requires testing hundreds of single-point mutations followed by rounds of combinatorial design, which can lead to many failures. This goes back to what I said in the beginning of the presentation. So they use PROS to design about 30 different variants of the spike protein. And the best variant they showed had 20 stabilizing mutations. And as you can see here, the functional yields rose by 11-fold. In this design. This is in hex cells. So this is something we've already seen before. But the really striking effect came when they tested the neutralizing efficiency of these vaccines. So they inject the same amount of protein into animals and extract the antibodies, and then they test neutralization. What you can see here is that the neutralization efficiency rises by 10-fold, an order of magnitude. And these designs are based on the Wuhan. So maybe this is still surprising, but what's more surprising is that this is maintained also for variants of concern, including Omicron, which is quite far from Wuhan. Why would that happen? Why would you get a higher level of neutralization efficiency from stability design, especially for these variants of concern, which are, again, quite far. So their hypothesis in this paper, which I agree with, but it is an hypothesis, it's very difficult to prove, is that improved foldability induces improved immunogenicity. So coming back to this plot, if you have a dynamic protein, and the spike is very dynamic, it can also lead to degradation, aggregation, and other problems with this protein, which would expose non-native, non-physiological surfaces on the spike protein, leading to the immune system developing antibodies that are not necessarily neutralizing for the physiological state of the virus. By contrast, if you have a construct that looks like this, then that gives the immune system more time to train on the correct physiological surfaces and produce a better immunological response. I find this to be very encouraging, and just thinking whether if this construct had been available during the first vaccination campaigns, maybe that would have spared us some of the chaos of these variants of concern. And also in this paper, they raised the point that maybe this could also enhance RNA vaccines, and this is something that we're testing now in collaboration, not on corona, but on another vaccine target. Let's hope that this works. So following all of these studies where we saw that computational design is now reliable enough to produce multiple variants that are highly effective, and these are automated strategies, we asked the question whether we could produce large numbers of sequences to explore sequence structure and function spaces very, very broadly. And by large numbers of sequences, I mean, like, let's say millions of different enzyme structures. So here, the goal behind combinatorial assembly and design of enzyme libraries, or CADENZ for short, is to design modular protein fragments that can be combined with one another to produce millions of full-length enzymes. So this was unprecedented at the time, but we were somewhat optimistic that modular assembly could work in design, because modular assembly is essentially the way that our immune systems generate the billions and billions of antibodies that we use to fight disease. So of course, our bodies are able to produce billions and billions of different antibodies, but there's no room in our genomes to produce so many antibodies. This is a trick question in admissions for students at Weizmann. So how does that happen? Of course, this is done by these V, D, and J cassettes in our genomes. So there are dozens of V genes, D genes, and J genes, which are randomly combined to generate specific antibodies when they're needed. And that leads to these many, many different specificities. So antibodies are really an amazing system for generating molecular recognition versatility. But outside of the immune system, we don't know if it's a similar system to generate versatile recognition. In enzymes, there's nothing comparable. So here we asked the question whether we could, inspired by antibodies, whether we could generate, using the same principles, a modular assembly of an enzyme family. So how would that work? We take many, many different structures, dozens of different structures in an enzyme family, align them, cut apart fragments from these enzymes. They're all homologous, as you can see, but there are also differences, including insertions and deletions in each of these fragments. And then design these fragments such that they would be maximally compatible with one another, and then generate full-length enzymes by randomly combining all of them, or essentially combining all of them all against all. How does this work in more detail? So here we targeted an enzyme family called glycoside hydrolase 10 xylanases. They break down xylan, which is a very large source of biomass similar to cellulose. So there are about 80 or so structures of xylanases in the protein data bank. We break them up into 4 segments like this, and then we use PROS to stabilize all of them, FunkLib to diversify the active site pocket, and then we developed a machine learning algorithm to look at all the combinations, all the design combinations of these fragments, and ask the question, which of these combinations tend to give you low energy, potentially functional, full-length enzymes? And then we simply order the best 30 yellow segments, green segments, blue, and red. If you have 120 such fragments, if you combine all against all, you get millions of these full-length enzymes. We clone them into yeast display and use and incubate this library with an activity-based probe. This activity-based probe doesn't hydrolyze. When it meets a functional enzyme, it irreversibly and covalently links to that enzyme. And because it's fluorescently labeled, you can then use FACS sorting and deep sequencing of the active site population tells you which were the active enzymes. And of course, in this case, we're interested in learning the rules for making functional enzymes. So we used machine learning in order to train an activity predictor based on sequence and structure features. And if this activity predictor is any good, we should be able to go back to the design step and design an even better repertoire. So what happened in this activity predictor? So we found 10 different structure and energy features that somewhat are discriminated between functional in blue and inactive designs in red. So for instance, and they do make sense. So for instance, the atomic density within the active site pocket or the hydrogen bonding energy within the scaffold of the enzyme. So they need to be good in order to find functional enzymes. But you can also see that these distributions are highly overlapping, right? So these would not serve individually as good discriminators. But by using logistic regression on 10 such features, we get excellent separation between functional and non-functional enzymes. So we applied this simple logistic regression model to generate a second generation repertoire. And here the hit rate of functional enzymes rose by tenfold relative to the first attempt at this. So this was really remarkable. It showed that this strategy, a learning loop, can really very efficiently increase increase both our understanding and capability in design. So the bottom line of this was that in 2 rounds of such design, test, learn, we obtained more than 15,000 functional and very diverse enzymes, encoding between 80 and 160 mutations from any natural enzymes. Oh, sorry. Yes.
No. I was just wondering. You have a logistic regression. Did you find like which features were the most important, particularly for that design? Was that consistent?
Yes. We didn't do so much feature importance here. We tried to get the best separation, and without using features that were correlated. So the way we did this is, of course, many other features were also exhibited some discrimination, but we eliminated features that were correlated with others such that we wouldn't be overfitting.
Oh, okay, that's good to know.
But you're right. This is a very good, in a different study, which isn't shown here, where we worked on GFP. We did feature importance and developed a slightly more developed machine learning. We used the random forest algorithm, and that allowed us to actually rank the features.
Okay, I have another separate question. How did you decide what was like a modular bit to?
How do I know that these are good fits? Yeah.
So like modular bits to your enzyme.
Oh, I see. Yeah. So this is based on a strategy that already Francis Arnold almost 25 years ago showed for recombination. So you look for sites within the active site pocket that when you align different structures, they all converge structurally very, very similarly at those positions. And that means that if you cut at that position, the dihedral angles would exhibit good fit.
Okay.
So we haven't automated it, but I think it's going to be easy to automate this. But by eye, it doesn't take more than 10 minutes to figure this out.
Thank you.
So a lot of diversity, but I'd also like to stress that this diversity isn't just surface mutations. In fact, what we see, this is 4 representative functional structures from this set. What we see is huge changes in the sterics and electrostatics of the active site pocket. And that suggested that there may also be functional diversity in this set. So in the second generation repertoire, we asked our collaborator, Herman Overclef, to give us another activity-based probe. So in addition to the xylam analog, we use a cellulose analog, and you can see that the molecular structures are similar but not identical. And in the second generation repertoire, we obtained more than 9,000 different xylanases and more than 3,000 different cellulases, and you can see that the overlap is rather small. So we are getting even functional divergence in this single repertoire. And this suggests to us, of course, we haven't shown that yet, it suggests that this strategy may enable discovery of non-natural activities. So generating such huge diversity in sequence and structure can enable maybe discovery of new functions. Do I have time to get to the climax of my talk? Yes, okay, good. This is really what I wanted to show. So going back to the opening slide, right, you remember that we had all these problems, low stability, low activity due to inaccurate design and inaccurate backbone design. But now we seem to have, so this was already 5 years ago or so, We thought that we had solutions to each of these individual problems: PROS for stability, FunkLib for accuracy in the active site pocket, and CADENZ for accurate backbone design. So we asked whether all of these together could give us high efficiency, new to nature activities. So this is still, it's in bioRxiv, but still not published. It's going to be published in a few weeks. We wanted to design a de novo activity that may be boring, but this would allow us to benchmark ourselves relative to previous studies. So we turned to the Kemp elimination reaction. This has been the target of de novo design for decades, literally decades. This is a very simple reaction, proton abstraction. A base on the enzyme abstracts a proton from a 5-nitrobenzoxazole ring. This causes a rearrangement in the ring, and this product is chromogenic, which means that you can very easily follow the kinetics of the reaction. And because this substrate is non-natural, there's no natural enzyme that has evolved a high efficiency activity for the substrate, and therefore we can't use a natural template for this activity. So we targeted this simple reaction, and you can already expect how this would work. Of course, we want to generate many backbones to generate a good fit for the substrate. So we use CADENZ for that to generate thousands of backbones for substrate shape complementarity. At the second step, we use geometric matching, which is an algorithm for 20 years ago to implement the thiozyme. And the thiozyme in this case is incredibly simple. It comprises a base to abstract the proton, and an additional aromatic residue to form stabilizing interactions with the aromatic residues of the substrate in the transition state. Keep this in mind. I'll come back to this aromatic group towards the end of my talk. And finally, we use PROS to stabilize the enzyme and FunkLib to pre-organize the active site pocket for activity. But the key problem here is that we can use evolutionary constraints outside of the active site pocket. There's no problem to stabilize the enzyme using PROS in that strategy, but we cannot use evolutionary constraint within the active site pocket because there's no evolved enzyme for this reaction. So here, Dina was the student leading this, excellent student leading this. Dina's idea was, okay, so let's just eliminate evolutionary constraints and just do this atomistically. I was very concerned about this. I wanted to use language models. I wanted to use a bunch of things. It didn't work. Dina was right. So the working hypothesis here is that reliable backbone sequence design would enable de novo design, that these were the key missing elements in previous generations of de novo design. So here, Dina designed 36 different Kemp eliminases. They showed, the best ones showed, we had 3 such designs. They showed the same levels of activity like 20 years ago. So fairly low, kcats of 0.01, kcat over km of 75. So 20 years ago, this would be news. Today, no. But now what normally people would do is take these enzymes through in vitro evolution. This would be rounds and rounds of evolution. 20 years ago, when they did that, there would be 17 or 20 rounds of in vitro evolution. Crazy work. Here, by contrast, Dina designed 12 variants using FunkLib with 5 to 8 mutations in the active site pocket. And this is the activity levels she got, 100-fold improvements. This design 27.7, almost 3 kcat per second, and almost 13,000 per molar per second. These rates are 2 orders of magnitude greater than any Kemp eliminases shown previously. So already, as Don Hilbert would say, respectable levels of activity. That's the best I could expect. The enzyme is also remarkably stable, 88 degrees melting temperature, and also cooperatively folded, which is very exciting. So again, we have stability under control. It's also, again, highly mutated, more than 140 mutations from the closest natural enzyme, including insertions and deletions. A crazy, very adventurous design. The active site pocket, at least in the model, seems to be to show a very good fit for the substrate, which would explain why it's such a good enzyme. We also crystallized the enzyme in the unliganded form, and you can see that the constellation of residues is largely as we designed it. So again, the trouble we saw in previous generations of design in terms of pre-organization in the active site, those again seem to be under control, except for the thiazyme aromatic residue. Again, keep that in mind. I'll come back to it, but very, very encouraging. So we have 13 designs here with 4 orders of magnitude differences in activity. So a really huge change, and 5 to 8 mutations from the starting point for each of these designs. So we can ask the question, do we actually understand in atomic detail why these huge improvements in catalytic rate? So here I ordered them by kcat over km, by catalytic efficiency. And what jumps to eye when we look at the energy terms from Rosetta is that the catalytic efficiency trends very, very closely with the catalytic base, aspartate 162, van der Waals energy. So why would that be? So the correlation coefficient here is 0.8, very high correlation. Why would that correlate so high? So van der Waals energy essentially means how packed is the aspartate with hydrophobic residues around it. And the greater the packing, the greater the pre-organization, and also the greater the desolvation, therefore the pKa shift. So the reactivity of this catalytic base would be higher thanks to this increased or decreased van der Waals energy. In addition to that, notice these 3 residues, 136, 183, and 216. So they trend from leucine valine to typically isoleucine valine, isoleucine. So these are very subtle changes, right? They're really, you wouldn't think this would be much, but if you look at the structure, compare the model structure for design 27, design 27.7, the improved version, what you can see is that these 3 residues really change the packing around this aspartate, leading again to greater pre-organization and greater water exclusion, greater desolvation, increasing the reactivity of this aspartate. In addition, another mutation from leucine to valine, this leucine forms a small steric clash with the substrate, and this is alleviated with this valine mutation. So again, we can explain why activity increases thanks to this mutation. And 2 other mutations at the opening of the active site pocket are from large and hydrophobic groups to smaller and somewhat more polar groups. And this is near the nitro, the polar nitro group on the substrate, adding to the desolvation, in this case, increasing solvation of the polar nitro group. So out of the 7 mutations in design 27.7, we have a good explanation for 6 of them. And it's a good local atomic explanation. And remember that here we're not using any phylogenetic constraints. This is purely atomistic design. So what I find to be very exciting is this is complete atomistic design of a high efficiency active site. There's no data, we're not using data, we're not using AI. This is purely based on physical understanding, which was, I think, the reason I joined the protein design community was to see something like this. So I was very happy with this. This is going to be the last data slide, and it's a little bit complex. I think it's very complex, so please bear with me just a little bit longer. So remember that the design protocol uses different components, but in a very sort of logical way, right? Process stabilize the enzyme, then FunkLib and other things to generate the active site pocket. So we asked the question whether these components, how do they play together? So here we sort of turn to an ablation study. What we do here, so first of all, the first line is design 27.7. To the left is the thermal stability, to the right is the activity. Then we go to the modular, okay, just consider these 4 designs to begin with. We eliminate the contribution for PROS from the active site and from another component, which I won't go into detail, and we're left with only the modular assembly. And you can see here, this is not unexpected, of course, that we lose activity completely and stability is also lower. If we only introduce the 11 mutations from PROS, we stabilize the enzyme, but still no activity. Again, no surprises. If we go back and eliminate the PROS mutations and only introduce the active site, we get a decent activity level. 2900 is pretty good, and the enzyme is still not so stable. But then when we introduce both PROS and active site design, what we see is that FunkLib essentially is optimizing stability and PROS is optimizing activity. We're getting higher activity than in the active site design alone, and we're getting higher stability than in PROS design alone. So this shows that these components are acting synergistically to improve both stability and activity, showing this is a good design strategy, something a good engineering strategy where the parts are actually synergistic with one another. So then Dina turned to the active site pocket and did an additional ablation study to see what are the contributions of the thiozyme residues. First, starting with the aspartate to alanine ablation mutation. Obviously, that kills activity, verifying that the aspartate is really key to activity here. And then the enzyme becomes, or it's no longer an enzyme, just a protein. The protein becomes just boiling resistant. It just goes crazy high stability, hyperstability. And this shows how this design protocol essentially maximizes the foldability and the stability of the protein. It also shows how negatively the burying this aspartate impacts the stability of the protein, right? One aspartate residue in such a hydrophobic region decreases the stability by so much. This is, I think, to me, it was very teaching. And then she next ablated the phenylalanine. Remember this phenylalanine that's giving us trouble. This phenylalanine was there from the first de novo designs of 20 years ago, again, to stabilize the transition state, the aromatic groups in the transition state, and also to delocalize charge. And so Dina asked Rosetta to give her another, she didn't want to go all the way to alanine. What is a different identity that would be stable and is not aromatic? And Rosetta suggested leucine. What you can see is that the activity, instead of dropping, rose through the roof, tenfold higher. So that shows that the aromatic residue was not just unnecessary, it was potentially harmful. And that was a huge surprise, right? Something that we assumed for 20 years was simply not the case. And this shows, I think, really very nicely how reliable atomistic design can test the fundamentals of biocatalysis. And what I hope is that this strategy will now lead people to study more complex reactions than the Kemp eliminates, right? Maybe we can move beyond that now. So just to summarize my talk, until a few years ago, computational design was not reliable enough to work on these complex, large and complex proteins, but this combination of AI modeling, atomistic, Rosetta atomistic design, and phylogenetic analysis is really giving us new control over proteins. The methods are reliable in general. For the first time, protein design algorithms are literally more effective than experimental screening approaches, and often no stability activity trade-offs are seen. And what I'm very excited about are these high throughput design approaches that I showed in CADENZ. We're now applying this to generate a universal antibody, human antibody repertoire for rapid and stable antibody discovery. So I think this may enable discovery of enzymes with activities not seen in nature, and also for antibody design. So I think exciting times for computational design. And thank my collaborators, the group, of course, and funding. Yeah, I took more time than I was allowed.
Sarel, thanks so much for an amazing talk. I will maybe take the privilege of being the host and ask the first question, and then I'll hand the microphone down. Yeah, stellar. One thing my mind immediately goes to is thinking about this thermal stability, right? And oftentimes the enzymes, right, we find from hypothermophiles are often really active, but their optimum reaction temperature is typically not room temperature. So just curious if you guys have explored different reaction temperature optima or tested these enzymes at different temperatures, and is there any correlation there you find of like where the T-opt lands?
It's a good question. I think it's when you increase the stability of an enzyme by, let's say, 20 degrees or so, it's fairly obvious that T optimum would rise simply because all activities rise when you increase temperature, right? That's a general feature because it's easier to cross the energy barriers. So I think that's a trivial result from enzyme stabilization. This is one of the reasons why in industry, for instance, they always look for a more thermally stable enzymes. But your question is somewhat different because if you take a thermophilic enzyme and put it in ambient temperatures, you often see much lower activities than you see in mesophilic enzymes. We don't see that for PROS. So you still see high activities in ambient temperatures, even though the enzyme has been stabilized.
Awesome, thanks.
Hey, Sarel, thanks for your talk. So kind of related back, my question is about like your very first point of that review articles, like, can we really make a naturally active enzymes? And can you discuss more on like the absolute kcat over kn value of all of this optimization? Because to me, like my understanding of when we make a new enzymes, like new to nature, is that because the original comparison, like wild type is already pretty terrible. So when we see like 100 or 1,000 folds of improvement, it's because at first it was not very good. But yeah, so I wonder like what's the absolute number here, and then in the distribution of natural enzyme kn, where does it fall?
This is it's an excellent question. If it's de novo, then you have no real comparison, but you can compare it to natural eliminases, for instance. Or in our case, what we did is a sort of just a general reference is to look at there's a paper from about 10 years ago from a colleague of mine at Weizmann Institute, Ron Milo, which looked at the distribution of kcats and kcats over kms in nature. And it's a bell-shaped distribution, but the median is about 10 per second for kcat. Our enzyme here was about 30 per second, so it's at the median. The de novo designs are typically 0.1. So the highest were 0.1, so 100-fold lower than the previous generations of de novo designs were 100-fold lower than the median enzyme in nature. So that means that they would be very poor enzymes compared to natural ones. They're really at the edge of the distribution towards the zero. Does that answer your question?
Yeah, then maybe next one is like, what do you think is missing? Like, what's the two magnitude differences?
Yeah, I think the 3 points I mentioned in the beginning, we literally could see this in the crystallographic structures of designed enzymes. The catalytic constellation was often off in the crystallographic structure relative to the design models. We observed that stabilities were low. And we didn't have control over backbone. So in order to achieve high shape complementarity for the ligand and you know for the thiozyme to be completely relaxed, let's say you have an aspartate and an aromatic residue, let's say you needed an aromatic residue as well, you want these residues to be completely relaxed when the substrate is there so that they don't need to rearrange, right? Because any rearrangement would cause the loss of activity. For that to happen, you need control over the backbone. It's unlikely that the natural backbone would give you exactly the geometry you need for that. So I think of the three algorithms that I showed, PROS, FunkLib, and CADENZ, were necessary for this. There may be other approaches for this, right? With AI, you can generate new backbones. I do think that natural folds give you more or better starting points for enzyme design than alpha helix bundles, let's say. But I could be wrong. Let's see.
Thank you so much.
Okay. Yeah, Sarel, thanks very much. This is a very exciting talk. So it was kind of the question that I wanted to ask as well, was sort of what's next. But where you mentioned like backbone compatibility, how much of that is also conformational freedom and dynamics? Like where does that fit into this?
Yeah, so dynamics has been brought up as a potential reason for why de novo designs have been so inactive. This mostly came up from the NMR groups who were looking at this. But I think that for these simple reactions like the Kemp elimination, all of the de novo reactions that have been tested were very simple. These actually require pre-organization. These are not cases where you need the enzyme to open and close. I don't think that dynamics are necessary. Of course, we don't encode dynamics. Everything we do is like to lock things down. And you see that we are reaching high efficiency. So I don't think that what we were missing were dynamics. I think the reverse, we were missing accuracy and actually rigidity. We did some MD analysis, or Lynn Camerlin, our collaborator, did MD analysis. She had done MD analysis on previous generations. And what she shows is the active site pocket, in our case, remains open and the substrate enters and leaves multiple times, showing that the active site is essentially oblivious to whether it's really pre-organized for activity, whereas previous generations would collapse and then the ligand can enter. So I think it's actually the reverse. In other more complex reactions where the substrate is large and where you need conformational change, that's a completely different story. To get there, we'd need to develop the methods much more. We're not there.
I'll take you back to the thermostability and the observation that you shared about improving stability correlating with improving homogeneity. That strikes me as quite profound, and also a bit in a contrast to something that has been reported and is still, I think, out there question. I think you would recall that the new looking paper was referring and testing immunogenicity of this completely de novo protein, and to my knowledge, it was hyperstable. It was about 100 degrees or something. So what do you think? Is there like some kind of a Goldilocks situation, which likely is the case, but de novo design proteins like the new looking were hyper thermostable.
Yes.
And supposedly the hypothesis there was that thermostability actually could decrease its presentation by enzymes not actually being able to chop it and present it at MHC2. So what do you think?
I don't know.
I'm trying to understand.
Is there evidence that proteases can't process these?
It's a hypothesis.
Yeah, I don't know. I think de novo designs are fairly small too, and that means that there are few peptides for presentation. So that may be an issue. I mean, not an issue actually. It's often a good property. But I don't know. I think that there's still limited experience with de novo designs in animals, and I think this is a really important question for the field of. Chris, you probably know more than anybody here.
Well, we also have a former Neolucan team member in the audience could probably answer as well.
Okay, yeah.
Jorgen, you want to speculate on the record?
On the
record. We have a couple of questions from our folks that are on the live stream, so I just want to make sure they get to participate as well. So excellent talk. How specific are these enzymes? Can you design for selectivity on substrates with high similarity? For example, design for activity on butyrate, but not acetate.
We worked on acetyl-CoA synthetases. Let me see if I remember correctly, and we did optimize for substrate selectivity in that case. So if this is a specific question about acetyl-CoA synthetases, then we have a paper on that in PNAS, I think 2017. If they just write me an email, I can send that. I can send that citation.
Great. The other question is, has this approach been used for improving developability of antibodies?
Yeah, okay, great. I didn't show any antibody data, but we are working very heavily on antibodies. We developed a method called QMAB for antibody humanization. Initially, it was for antibody humanization. It's based on similar principles of optimizing in QMAB, we're optimizing the native state energy as usual for us, but subject to using V and J genes from human sequences. So even if we start from an animal, regardless of whether we start from an animal or a human antibody, we use V and J gene sequences from humans. We shuffle all of them computationally and look for the lowest energy combination, given that the CDRs are as in the parental antibody. That was the summary of QMAB. And we have seen that improves developability. What we saw is expressibility often rise. We maintain affinity, so expressibility, stability, but also more, I'd say, softer or long-term developability issues. So for instance, polyreactivity, this was done in collaboration with Pete Tessier's lab at the University of Michigan. We reduced the polyreactivity of an antibody that he was working on in a case where there was tryptophan oxidation, at 3 a.m. now, tryptophan oxidation, which leads to degradation of antibodies. We could also improve that property, even though we're not touching the CDRs in that case. And there was a few other such qualities. I'd like to say that we don't have a complete solution to antibody developability. There are some properties that we've struggled with, like self-association, sorry. But we're working on that. If anybody's interested in collaborating with us, we're really looking for collaborators on that.
Great talk. So I have a question about pH. So in your designs, for example, in the enzymes where you mutate in constellations, sometimes these residue need specific pH environments. Do you take that into account in your design, and how?
We're using the Rosetta energy function, so that isn't really directly taken into account. But if you think about it, when you use PSSMs again, evolutionary constraints, we're not going to introduce mutations that are never seen in evolution. And I think that probably keeps us safe from making mistakes or treading in regions that we shouldn't tread in.
Thank you.
In the section on modular assembly,
Yes.
there was a question about how you decide where to cut. And your response was something about in the conserved areas, the dihedral angles would be constant. And that Francis Arnold had discovered this. Can you elaborate on that? Why
Sure.
that's the case and how
Yeah.
constant are those dihedral angles?
So the method that, Francis Arnold developed was called schema. And this is called, I think, structurally conserved. I'm not sure now with the naming, but the principle behind this is that if you think, what we're doing is essentially splicing together fragments of the enzyme. And for that to work, the dihedral angles coming in and going out need to be exactly right? Because even if you have a slight change, we found that even 0.6 angstrom RMSD already gives you really weird angles. So we're really strict on that. But if the angles are good, then you can practically recombine all against all, and you know that you'll get a good peptide bond or an appropriate peptide bond in that connection. So it's sort of a way to splice together fragments of enzymes while being relatively safe. If you did the same thing in a more variable region of the protein, then the dihedral angles in a fragment coming from one homolog would not be a good fit for another homolog, and then it gets strained in the peptide bond and probably very poor folding. Yes.
Can you talk a bit about the computational complexity? Is it a concern at all?
Yes. For PROS and FunkLib, it's very easy. We open this to outside users, so that means that we're not concerned. For something like CADENZ, it's very expensive because we're enumerating huge spaces of conformation space. Now we're developing a method based more on AlphaFold than on Rosetta. AlphaFold is doing these splice operations in Rosetta was very, very troublesome. In AlphaFold, it's just you essentially do this bioinformatically, right? You combine sequences together and you fold them in, and you get the structures. It's still high, it's still demanding, but it's okay.
I'm surprised even about PROS because you said you make up to 30 mutations in one design. The combinatorial complexity of that is huge, right?
But we use Rosetta. Essentially, Rosetta Packer can handle such large spaces. And keep in mind that we're also limiting the design sequence space tremendously using PSSMs, for instance. We're eliminating any mutation that's rarely observed in evolution. We had this calculation in the original PROS paper. We were working in acetylcholinesterase. This is an enzyme of 550 amino acids. So the theoretical complexity, if you just applied standard Rosetta calculations, would be on the order of 10 to the power of 715. That would be very difficult. But after these prunings, evolutionary and energy prunings, you get down to a space of 10 to the power of 31.
That's nothing.
That's still bad. But Rosetta has been able to handle such spaces for many years. So yeah. We're not enumerating, right? So it's the Packer does this more effectively using Monte Carlo simulations. Yes, sorry, yes.
So for enzymes, are you able to or interested in modeling cofactors or some of the next steps of?
Excellent, yes. So given AlphaFold 3, we can model cofactors very easily. And this is something that and not just cofactors, actually substrates as well. So what we're looking at now, and we're doing this retrospectively, we're looking at previous FunkLib design runs from our lab and from others who have published data, and trying to correlate what we see from AlphaFold with the experimental results. And in some cases, we're seeing good agreement. These are the cases where we see large improvements in activity. When there's large improvements in activity, the AlphaFold models give you near attack confirmations. And that's, I think, very promising. We're still not absolutely certain about this, but I think that maybe not in the distant future, we'll be able to direct FunkLib for substrates rather than just saying, here's a panel, 50, go test it. We may be able to direct FunkLib to specific substrates.
Thanks.
Hey, so you're using evolutionary constraints. So it's cool that this works in like E. coli and very distant organisms.
Come again, can you speak closer?
You're using evolutionary constraints to increase stability. So it's cool that it works in like distant organisms like E. coli and everything. Is that always the case? Usually the case? Are you limited by the breadth of your tree that you're able to sample? Or?
So the RH5 example, the malaria example, was we thought this would not work. We were right because there were so few homologs, but it did work in that case. So it shows that we need some information from nature, but maybe not so much. And again, I mentioned that in one recent case, we even used MPNN, protein MPNN to generate PSSMs, and that in that case at least worked too. I don't know if this is general though, right? Because, but if it is, then maybe even orphan proteins could be done this way. Maybe even de novo designs. I don't know.
So when you were describing the FunkLib pipeline, I believe you said that you would do the virtual screening and the down selection process without the substrate. So, and then you would do the experimental validation. So do you have strategies right now to deal with modifications to the organophosphates?
Modifications to the, so if you generate Novichok, okay,
exactly.
We think that they're active against Novichok as well, if that's the question.
Awesome. Sarel, thanks so much for an amazing talk. Great Q&A session. Let's thank Sarel one more time. All right, so we still have a lot of pizza and beer. We don't want to have to carry this or deal with it, so please come hang out, ask Sarel more questions, and we'll see you next month over in Cambridge at MIT. Cheers.